Okay, I think we'll go ahead and get started. We got 90 some participants. I'll take a few minutes to give an intro, and this will be recorded and put up on YouTube probably later this week. So welcome everybody. I'm Sam Burden, assistant professor in electrical and computer engineering. This is our departmental colloquium. It's my pleasure to host and introduce Professor Noah Cowan, who's a faculty in mechanical engineering at Johns Hopkins University in Baltimore. So Noah did his BS in Ohio State, his MS and PhD at University of Michigan. Then he was a Miller, very prestigious postdoctoral fellow at Berkeley for a couple of years before he started as a professor at Hopkins, and then he's been there ever since. He's won a number of awards. He got the P case, so he got to shake Obama's hand in 2010. I think we're all a little jealous of that. A McDonald Foundation Award for Complex Systems, some excellence awards from his university for teaching and kind of contributions to community. My kind of take on Noah is somebody that I've looked up to and whose work I've closely followed throughout my career. I was kind of first introduced to his work when I was an undergraduate researcher in the lab of his PhD advisor, Dan Kodacek, who also happens to be the PhD advisor of our department chair, Clavens. And interestingly enough, to me at least, who I believe overlapped with Noah a bit at Michigan. Yeah. And what I think is really interesting about those two examples is they're people that started out in robotics. I mean, Dan Kodacek is a roboticist through and through, kind of a pioneer and a leader in that field. And yet both of them kind of took the training that they started with and then moved into completely new fields. So not content with the nascent emerging discipline of robotics, they took those techniques and their knowledge and moved into completely new directions. In the case of Noah, he's become a full-fledged card-carrying neuroscientist, but with whose core training and interests are in the development and application of control theory techniques, and so that's what we're going to hear about today is his use of these techniques, studying all manner of different neurobiological systems, animals, people, and different things like that. And so with that, I'd like to turn it over to Noah to give him the floor. So let's give Noah a warm Zoom welcome. So we'll go like this.
Thanks so much, Sam, for inviting me. I love UW. I did a sabbatical there many years ago, and there's so many great people there. It's such a great place. I wish, in fact, I'm really especially sad that this is this year that I was invited back to give a talk. I've never given a talk, I think, in ECE because I love to come there so much and just couldn't do it. So I'm really, really delighted to have been invited. I did want to set the record straight on one thing. I didn't actually get a Miller award at Berkeley. I wouldn't really care to say that, except that this is recorded. I want to kind of set the record straight, but thank you for that very kind introduction. I think everything else was basically right. So this is a little bit of a cheeky title, Neuroscience in the Matrix, but I'm going to talk to you today about how we've used control theory both as a framework for understanding the problem that the brain faces in controlling movement, but also applying the tools to wrap feedback loops around behaving organisms in order to both modify their performance in some cases, but also to help us gain even deeper insights into the controllers that are in the brain itself. And we do this in my lab in a number of model organisms, starting with my work as a postdoc in Bob Full's lab, studying the lowly cockroach here, seen following along a wall at many body lengths a second in complete darkness, and very ably sort of tracking that corner. In fact, I think you might be able to see the Journal of Experimental Biology cover there on the wall. My first foray into biology, trying to understand everything from the mechanics of individual sensilla on the cockroach all the way up to the whole problem that the nervous system is solving in doing this high-speed locomotion. Human movement and its control with Amy Bastian. One of the topics that I really have loved lately actually has a lot of overlap with Sam, which is using feedback control theory to understand human movement and its control in upper arm reaching. That's another project that we have with Amy and also Brent Gillespie and Jim Frudenberg at University of Michigan. We just had our first, one of our first control papers came out with Amy Bastian just 2 days ago in eLife. I won't be talking about that today, but that's a fun study. My most, the most, I say, core model system that we study in my lab is the weakly electric glass knifefish. I'll be checking on my fish as soon as we wrap up with meetings this afternoon over in my lab. And here you see the bottom view of an electric fish that's moving back and forth to stay hidden inside of a tube. You have to keep in mind that this fish is quite small and it's hiding in this tube because there's a hungry graduate student standing over the tank that it's trying to stay hidden from. And so it follows back and forth inside that refuge to stay hidden. And then some work that I was really delighted to do when I visited 10 years or so ago with Tom Daniel, looking at visual abdominal reflex response in the hawk moth, among other organisms that we study in the lab. And in all of these systems, we take a control theory and system identification approach where we think about the cascade of information from sensory systems through the central nervous system out through the muscles and into the mechanical system, be it sort of just arm movements or whole locomotor mechanics. And then, but ultimately the largest source of sensory stimulation that most animals receive on a regular basis is caused by their own movement through feedback. So when you turn your head, there's a large retinal slip that you experience that's caused by what biologists call reafferent feedback, or just simply closed loop feedback between the animal's nervous system and the environment. And certainly it's really interesting to think about the problem of an object that comes into your environment and catches your attention, but thinking about the nervous system as a feed forward entity is just kind of not the right way to look at it. The nervous system can really only be understood, in my opinion, when thought about in a closed loop context. And so we take this kind of closed loop view, and then we do as many things to the behaving system as we can in order to sort of tease apart both the individual components of this feedback system as well as their contribution to the overall closed loop control, whether it's moving visual or other sensory scene items, changing the visual or sensory conditions by changing the lighting, or in the case of electric fish, changing the conductivity of the water, changing the locomotor mechanics by modifying the substrate that an animal is running on, changing the flow conditions and so on, providing mechanical disturbances, and even opening or closing feedback loops. And then while doing that, taking measurements from various points in this diagram. This is obviously a very simplified diagram, but taking measurements, electrophysiological measurements, EMG measurements, those are electromyograms measuring the neural activity, the electrical activity in muscle, measuring the forces that those produce, measuring the overall kinematics of the organism. And we've a number of review papers where we've discussed this kind of overall approach. Today, I'm interested in taking this a little bit of a step further and actually closing a feedback loop around this, these behaving systems to try to interrogate their performance. We're going to start with this weakly electric fish. This is the weakly electric fish Eigenmania virescens. It's also called the glass knifefish. This is a joke that never gets old to me. I think that it's God's gift to control engineering because it has eigenvalues in its name. It's the Eigenmania virescens is the genus and species. And these fish actually are remarkable organisms from a sensory motor control perspective. It's really just sad that I can't tell if you're not laughing or it's just that everybody's muted. Either way, it's awkward.
We're laughing.
So awesome for studying from a control theoretic point of view. Well, one is that they have a visual system like all or most teleost fish have, but in addition, they have an electrosensory system that operates as a parallel non-contact sensor that maps the three-dimensional world onto a two-dimensional sensorium that is physically independent from the transduction of light information. And so it allows you to do things like create sensory conflict of non-contact movement in order to look at multisensory integration. But then in addition to that, they have this ventral ribbon fin that allows the fish to swim nearly equally well forward and backward and allows it to kind of hover in place inside of say a moving refuge like I showed at the beginning. And so through this mechanical and these sensory and neural adaptations, it allows us to probe deeper into the closed loop control. And so weakly electric fish is not necessarily something that's that common of an everyday thing. So let me give you a little bit of a background on the biology of these organisms. So they're widely studied in their field of neuroethology, a well-studied organism for looking at the integration of neural systems and behavior, because their primary mode of sensing, which is not their primary, a primary mode of their sensing is through the generation and detection of an electric field. And so it makes it very amenable to laboratory analysis, because you can just literally put a pair of electrodes in the water, which is just, put a set of wires down in the water and hook it up to a Radio Shack amplifier or a sophisticated recording device and listen to their electric field by amplifying and playing it through a loudspeaker, because it's in an audio band. And so you can measure and manipulate that electric field and do experiments. And so that was one of the main roles of this system for many years, was to look at how the animal modulates based on ongoing sensory feedback. How does it modulate its electric organ output? We use it for another reason, which is for this locomotor control. And so if you took a sort of a sketch, made a sketch out of took a sketch out of Heiligenberg's book of what the electric field might look like, these would be the current lines generated by the electric dipole that the fish generates. It's an oscillating electric dipole, and at any moment the current would be flowing, around objects in this round. And this particular object, if you can see my cursor, would be, say, an insulating object, and it would also distort the electric potential and create a change in the voltage on the surface of the skin, allowing the electroreceptors to detect that and therefore sense that object. Malcolm McIver once described this in a really elegant way. He thought about this as touch at a distance. The animal can, this isn't a very good long range sense, but for objects that are in the immediate surround, they can sort of feel where they are. Maybe much like, you might feel a static object getting close to your arm and your hair standing on end, is the best analogy I can make to anthropomorphize what they might be experiencing. So as that object moves, or in this case, as this tube moves, the electric fish is able to sense that motion and control its position inside the tube based on the combination of electrical, visual, and maybe tactile feedback. Can feel the movement of the water, for example. And I like this video. This is a really old video with Eric Fortune moving this tube back and forth. And when I watch this video, even to this day, it looks to me like that fish is attached to the tube with a spring and a damper. But the only thing attaching that fish to the tube is its own neural feedback. And so we obviously thought Eric's arm would get tired if we did that, if he had to do that all day. So we set up a simple computer-controlled version of that, cut off the bottom of the PVC pipe, and now we're videoing from below. And you can see these really nice pectoral fins. Hopefully they're coming through. Is the video quality okay? Okay, good. You can see these nice pectoral fins and this undulating ribbon fin. And the tube is moving back and forth, and the fish is moving back and forth. And our goal is to try to figure out the control system. A lot of this work was, well, here are all the people that have contributed in various ways to this process. Eric being my longtime collaborator. So we've spent many years investigating the interplay between the controller and the plant. And so let's just say, for the sake of this talk, that we solved that. We know what the controller is and we know what the plant is. I can leave you to look at some of the papers for how we figured out many of those things. But then something really dramatic happens when you turn off the lights. When you turn off the lights, so you see two videos here that are exactly the same, in fact, taken in the same, within a few minutes of each other. In the top video, the light is on, indicated by this icon here. In the bottom video, the light is off. And then in a moment, we turn off the light in the top video and turn on the light in the bottom video. And so you see a dramatic difference in the behavior of the animal. When the lights are on, the fish is just tracking very smoothly. And when the lights are off, the animal's like it's feeling its way around in the dark using the electrosense. So I'll let that loop for a minute. And so this idea of vision modulating electrosensory active sensing behavior, what we think is happening, and I'll give evidence for this, is that the fish is feeling its way around to basically excite the electroreceptors in its skin. Something like, if I give you an object and tell you to sense its texture, you'll rub your hand on the object. If I tell you to sense its weight, you'll move your hand in an up and down motion. We perform these motor behaviors with the goal of extracting sensory information. And what we think is happening here is that the animal is moving back and forth and probing around in order to improve its electrosensation. So why is it doing that? So in this first little silly example or silly illustration, we're going to close a feedback loop around the animal and try to figure out what's going on. So this was work primarily by Dev Jyoti, who's a PhD student here at Hopkins. He did a research rotation in my lab and actually got a really nice paper out of it. Luke Aaron was an undergraduate researcher over the summer, and he did another primary bit of the analysis. And then these were also all contributors to this project. The primary work is Dev Jyoti and Luke. And what we did was we said, okay, let's take the animal's movement. Let's measure that in real time with a video camera. Play that, use that to modify the location of the refuge in a closed loop. And note now that the consequence of the animal's movements is still in direct control of the animal, but now through different feedback dynamics, not just the feedback dynamics of its own mechanics, but also because we're modifying the motion of the tube. For example, if the fish swims forward, we might move the tube backward in response, amplifying the error signal that it would have otherwise received. Or if the fish moves forward, we might move the tube forward, but at a smaller gain, attenuating the error signal that it might receive. And so this is a little bit out of order. I should have showed this figure sort of before I showed the last figure, but just to review the experiment that we just showed, as this is just straight up digitization of the video that I showed a minute ago, where we start with the lights on, and you can see the fish is tracking quite well. And then right at the moment that we turn the lights off, the fish begins exploring, but you'll notice that on average, it's still tracking the tube very well. And vice versa, if the lights start out off and then we turn them on, the fish sort of locks in and starts tracking well, but before that it's doing this active sensing behavior. Why would you do that? Well, I'm gonna challenge you to try your best to focus on the black plus sign in the middle of your screen and not to move your eyes around in a saccade. And I'm gonna press the play button. And I'm going to do the same thing. And as you do that, you should notice that the purple fades away. And if you look around, you'll suddenly see it again. But as you're focused, depending on if you've got the scale right and you're the right distance from your monitor and so forth, the purple will begin to fade away. And that's a process known as perceptual fading. Hopefully most of you got to get that. If not, look up Troxler fading on Wikipedia, as you see from that image there that I downloaded from Wikipedia. And you can play this for yourself and mess around with the scale until you get it to work. And so the idea is that if you don't actively move your sensorium, you adapt to the sensory stimulus that you're receiving. It's the same reason that you can't feel your underwear right now. You adapt to the sensory stimulus when it's persistent, right? And so you could do other experiments like, put one hand in warm water and one hand in cold water and let them sit there for a while and they'll begin to feel the same temperature. We're really good at detecting change. We're not so great at detecting absolute. And so what we think is happening, and this is an incredibly crude illustration, what we think is happening is that see how this video works, that the animal is moving back and forth so that the pink dots that are on the surface of the skin, which are my very crude representation of the electroreceptors, are receiving changes in their electrical feedback, constantly stimulating the electroreceptors. And in a very simple nonlinear model of the electroreceptors, if we take the simplest possible plant model, which is just a mass damper written here in state space form, and we say that the output is the time derivative of a set of sensory measurements, then the resulting system is linearly unobservable in a neighborhood of an equilibrium point. Meaning that basically the obvious, if you're successful in tracking and you stay stationary relative to the object, then you cannot detect slow drifts of your position relative to that object. So you have to therefore begin to move to re-excite that sensory stimulus, re-excite the sensory receptors and get them detecting change again. And so local observability is sort of only guaranteed if your velocity, the second state variable, is non-zero in our simplified model. So the fish has to keep moving. So, well, that's our theoretical framework for thinking about this, but is it really borne out by the biological measurements? Like, that's my hypothesis, but it's not enough to say, okay, that must be true because we have a model that says that that would work. So we have to do a test. So the test that we did, the first test that we did, was we modified the conductivity of the water and then measured whether or not that had an effect, all other things being equal, did that have effect on the active sensing behavior? And so we did that in two conditions. What you're seeing here is the, on the x-axis is we basically took the discrete Fourier transform magnitude of the fish's movement across a low range of frequencies, and plotted that Fourier magnitude as a function of the movement frequency in different conductivities and in different lighting conditions. And so when the lights are on, the animal's not moving very much, but you see a slight trend upwards in the total amount of movement amplitude as we increase the conductivity. And what does increasing the conductivity do? It basically shorts out the electric field, you can think of it that way, and it decreases the electrosensory saliency. But on the other hand, when we turn off the lights, and we know that really excites the active sensing behavior, or at least this putative active sensing behavior, this thing that I'm calling the active sensing behavior, as we then crank up the conductivity, you see a much more substantial increase in the movement content, if you will, of the animal in response to that change in conductivity. So we don't just think it's purely a light dark, like, hey, the lights are off, it's time to party, I'm gonna move around a lot. But the fact that they modulate the amplitude of those movements due to a change in the conductivity is highly suggestive that it really is in order to gain more sensory information. And so, so we published that, but we wanted to dig a little bit further and try to figure out what exactly they were controlling. So this is where now the closed loop experiment comes in, where what we did was we took a measurement of the fish's position, multiplied it by a feedback gain, and played that into the shuttle movement. And then in addition, when the fish swims forward relative to the shuttle, obviously it's as if the shuttle is going backwards, so we get a negative gain to self-movement in the absence of this feedback. Here's just a little simple schematic illustrating the setup that allowed us to do this. We have a mirrored position below, the fish is in the tube, and then we can measure its position. And we're illuminating in infrared so that we can see the fish in the dark. And then we take this green dot's measurement and then feed it back to become the red dot. And so I'll show you an example illustration of how that works. So the top video is going to show you the lab fixed frame, like the video camera's recorded view of the fish and its position, and the shuttle in its position. And you'll see that the fish moves equal and opposite to the shuttle, because we've just prescribed the shuttle to move equal and opposite to the fish. And then we're going to take the fish-centered point of view and show you what the fish sees at the same time. So there's this initial ramp up where there's no feedback gain turned on. This is a technical detail that I can go into if you're interested, but by the time you hit 10 seconds, the fish is in complete closed loop. So you see the fish move backwards and the shuttle move forwards. The shuttle position is illustrated in red, the relative shuttle position, and the fish position is illustrated in this greenish color. And then the initial reference point is the blue position here. And then what the fish is seeing is an amplified error signal as a result of its own movement. That's what this video is demonstrating. And you can do the opposite as well, which is we can, we'll skip this little ramp up period and jump into the closed loop period. When we move the tube in the same direction as the fish, but at a lower amplitude, then the fish sees an attenuated error feedback as a result. So I'll let that play for a second so you can get some intuition for that. Okay, so so what are we doing there? We're taking this, we're looking at how does the fish modulate its behavior in terms of this gain? And our basic idea was, if it needs to maintain a certain amount of error signal in order to do good sensing, then when we amplify the sensory feedback, it ought to attenuate its movement. And when we attenuate the sensory feedback, it ought to amplify its movement so that the error signal, say root mean square, or the error signal's error distribution ought to stay roughly unchanged. And so here's again an example trial showing on the x-axis time and on the y-axis the position of the fish in green and the refuge in red. And of course, they're equal and opposite because we made it so. And then we, many of you may not know what this device is, but it's a cassette tape. Then we took a recording of this. The shuttle did something here in red. We simply take a recording of that and play it back in open loop. And we now the fish has no control over the trajectory that the shuttle is generating, because we're taking a previously recorded shuttle trajectory. And what we're going to look at then is, what does the fish do in response, right? And of course, we don't expect exactly the same thing, but what we want to find out is, what are the quantitative and qualitative differences between what the fish does when it's in control of its own sensory feedback and when it has less control over that feedback. And so what we found, if we take the root mean squared movement of the fish, when we augment the feedback with either a small positive gain or a large negative gain, then we see that the fish's movement in the open loop replay is not the same as when it's getting the feedback. And so let me parse this a little bit more. If it were the case that the fish was moving roughly the same amount, irrespective of, it's getting, okay, so the fish is getting exactly, in this experiment, let's say right here, the fish got exactly the same shuttle movement in both open and closed loop, because we take the closed loop trial and replay the same trajectory. But when the shuttle's position is being attenuated, the air feedback is being attenuated in closed loop, the fish is actually moving more in this experiment when it's in closed loop than it was when it was in open loop. The open loop is the y-axis. So when we attenuate the fish's movement, which are these orange dots, in feedback, the fish moves more in closed loop. And when we amplify the fish's movement in feedback, the fish moves more in open loop. Whereas if we look at the air signal, that is the difference between the refuge position and the fish position, they collapse to being the same. So the fish is always in these experiments generating the same amount of air feedback in the two conditions. That's the main take home. This is a little bit easier, this collapse is a little bit easier to understand and interpret. All right, so gamma less than 0. I think I have this backwards. Gamma less than 0, this is backwards. Gamma less than 0 attenuates, sorry, I'm wrong. No, I had it right the first time. Gamma less than 0 means that we amplify the feedback. That means the tube moves equal and opposite and amplifies the feedback. Gamma greater than 0 means that when the fish swims forward, we make the tube move with it and we attenuate its feedback. And what we see just quite clearly in these example plots that illustrate the point, when we amplify the feedback, the fish moves less. You can see just from the red wiggles. And when we attenuate the feedback, the fish moves more, right? And you can see that over a, if you again go into the air coordinates, that the air distribution is roughly constant among those trials. And so this is a technicality, but if you do a change in gain to the right coordinates, then we actually expect a linear plot as a function of gain for the RMS movement that the fish is actually trying to maintain constant sensory slip. And when the lights are on, we see that we follow that linear trend. When the lights are off, there's kind of an attenuation at the highest gain values. And that has to do with the fact that when you're forcing the fish to move a lot with these high positive gains, what you're doing is probably reaching a trade-off between sensing efficacy and effort. And so we end up getting a slightly sublinear trend here. This is for an individual fish, and this is collapsed across fish. But by and large, we see that the air signal is quite close to constant for these animals. So one of the things that I got quite worried about when doing these analyses is that maybe you could explain this change in the active sensing is just a mere consequence of the exact same controller with the exact same active sensing, but when you actually close a feedback loop, you modify the dynamics. And so if the fish has a feedback controller together with this open loop active sensing movement, if it was purely open loop, then obviously the effect of that active sensing movement would get fed back and would be affected. I've put the two pathways, the gamma and the -1 in the same, I've collapsed them here into the same feedback. You would expect that A of t would get shaped by the modified closed loop dynamics. And so I won't go into the details of these graphs, but were that the case, you would actually expect the estimated frequency spectra of A of t to be constant. If it was the case that the fish were not modifying its active sensing behavior. And so this is just a control, if you will, to double check that it's not just a consequence of the filter that we've developed by adding this feedback that is actually shaping the performance of the animal, and that it is actually actively modulating this A of t. And it's doing so by, over an order of magnitude. All right, so what did we see from this? So first of all, active sensing movements do change as a function of stimulus saliency, which makes sense. That was our earlier study. What I find interesting about this is kind of the lack of separability. In engineering, we like to think about, we have sensor system, we send that through a state estimator, and then we use that to inform our controller. But in biology, that's almost never the case. The animal has to reach out through its biomechanical plant in order to modulate the sensory feedback that it receives, violating this notion of separability that we take for granted in engineering. And then by closing a feedback loop around the animal, we allow us to interrogate the goal of the system, which is to maintain this sensory slip at a near constant value across conditions. So in the second example, and I set my watch here, okay. In the second example that I want to show you, we are looking at path integration in the integration between path integration and visual landmarks in the mammalian brain. And so, first of all, broadly speaking, navigation refers to the idea of figuring out where you are and what course you need to take in order to get from point A to point B. And you can look at this in a wide range of biological systems as well as from an engineering perspective. My colleague here at Johns Hopkins has done really pioneering work on how to do deep sea underwater navigation, for example. And so I guess I had little videos here that I needed to animate. But animals, including humans, do navigation as well. And in particular, I'm going to look at the mammalian navigational system. And so the idea is that in rats and other mammals, there is a representation of the animal's position in space in an area of their brain called the hippocampus. That's not so important what it's called. And the way we can examine these hippocampal representations is by implanting, this is a hyperdrive, and it has a bunch of electrodes in it that you can turn a little lead screw to position them in this very particular place in the brain, as illustrated here. This is the hippocampus, this curved structure here. And then as the animal performs a navigation task, this is a top view of a rat running around in a square box. And what we're seeing is every time a particular place cell fires, that is the neural activity of that place cell gives a spike, then we put down a red dot. This is not our data from our lab, by the way. And you can see that this particular so-called place cell selectively fires when the area is in this region of the box, okay? And it's kind of a remarkable finding, in fact, led to the Nobel Prize a few years ago. Place cells, along with the discovery of another set of cells in the medial entorhinal cortex called grid cells, formed the basis for the Nobel Prize, I think in 2016 or 17, I can't remember. And then what you can do is you can take the aggregate firing for that place cell and estimate a firing rate map. And so this would be this area that's a hot color would be called the place field of that place cell. So this allows us to therefore interrogate, and this is the Mosers, Edvard and May-Britt Moser and John O'Keefe, who won the Nobel Prize for discovery of cells that have this kind of selective encoding of space. And what's important, actually, I want to say, is that what surprised me as an engineer is just how robust a finding it is to see that there are these allocentric representations of space in the mammalian brain, that the animal has kind of a three-dimensional worldview of the environment, three-dimensional map of the environment, not three-dimensional, an allocentric map of the environment, and then can position itself within that as measured by the activity of these spatially selective cells. And so how does it actually achieve that? Well, this is a really simplified cartoon model, but it has an internal representation that takes into account a wide range of sensory cues. And I should also want to mention that this area of the brain is pretty far removed from the sensory periphery. So it's thought that this is an internally generated representation that is kept in constant congruence with the sensory information that it's receiving, but it's not a direct readout of the instantaneous sensory values. So for example, if I have a set of place cells that are active when I'm at this position in my office, if you can see my video, if I turn and look over now at my chalkboard or look at my bookcase, those place cells would still by and large be active, even though I'm getting very different sensory percepts at that moment, because it really is an allocentric representation of space and not an egocentric representation of space. In addition to these landmark-based cues that I'm using to orient myself in a room, I'm also getting a set of cues whose instantaneous values cannot tell me exactly where I am, but that I could integrate over time. Say they were velocity values, or velocity and or acceleration values, but I'll say in a simplified way, I can integrate those over time. If I know my velocity vector over time, or a differential displacement, I can then estimate a differential displacement in my cognitive map, right? And I would do that by some sort of cognitive gain, we'll call that H, okay? And then I would integrate that cognitive gain as a function of my differential displacement to figure out how to update my internal representation, okay? If my gain were too large, I would move my internal representation too far. And if my gain were too small, I wouldn't move far enough in my representation. So to do a thought experiment, if I stand in my room and I close my eyes and I take 2 steps, when I open my eyes, I'm not surprised about where I am. I performed some sort of internal path integration and I updated my position in my map. And then I had an expectation of what objects I might see in that new position. But if you do this experiment over a large room, you'll find yourself reaching for the wall before you get there because you might be overestimating how far you went, okay? And you may have different gains for each of these different modalities and so on. Well, in order to interrogate this interplay between path integration and landmarks, my former PhD students, now postdocs, and actually Manu Madhav is going to be starting a faculty position at University of British Columbia in January, built this pretty remarkable virtual reality apparatus where the rat runs around in circles on this table, and it's inside of a planetarium-style dome, which has objects projected on the interior surface, objects or stripes or other things that we can project on the interior surface, and then we do manipulations of that in order to interrogate the interplay between path integration and landmarks. And so, so this is our rat, the protagonist. Here's a video in the dark. You can see of the rat picking up chocolate milk as it runs around on the table as its reward for exploring this environment. Show that for a few minutes longer. And you can see, actually, here's the hemispherical mirror. You can actually see with the regular camera, you can see the projection of the objects on the wall. The current view isn't letting us see those objects, but they're being projected up onto the side, onto the sides. There was a little glimpse of one. There's the cheese slice right there. So, so here is the bird's eye view. And what I'm going to show here with these blue dots is one place cell activity, the activity of one place cell as the animal runs around in circles in this dome. And you can see, because we can see the reflections of the objects in this hemispherical mirror, in this front surface hemispherical mirror, there's also this ring of light, and that's a technical detail, but we keep that ring of light present at all times so that there's always a nice contrasting cue on the ceiling. So when we turn off the landmarks, we're not going to turn off that annular ring of light. But anyway, you'll see these landmarks are here, and this place cell is firing consistently in a particular region of the space. Okay. And so now, what's our manipulation? First of all, what's our question? Our question is, this hippocampal gain, H, that we're calling it, is that a variable that the nervous system, well, first of all, is this even a good framework to think about it? Is it reasonable to think about the idea that when we turn off the landmarks, we have some internal gain, number one, and is that gain a manipulable plastic variable? Part of this was inspired by a really interesting paper that I read when I was a fresh PhD student just starting in Bob Full's lab. There's a paper that was coming out on looking at ant navigation. And there were these desert ants that were trained to forage for food at a certain distance. And then what they did was they took the ants and they did two manipulations and a control. They took one set of ants and they put stilts on them, and this in complete darkness. And those ants traveled too far to look for food. Then they took another set of ants and they cut off their tarsi, the last segment of their legs. And those ants didn't travel far enough. So they were obviously doing path integration based on a knowledge of sort of how long their legs were, so to speak. And then the control group was like Goldilocks and it was like the baby bear, I should say, and searched just right for the right distance for the food. And so our question was, well, can we do something like that, a little less dramatic, and create virtual stilts and virtual short legs by manipulating the gain as a function of the animal's movement in the environment? So if we could make the animal feel like it's running really fast by counter-rotating the objects as a function of the animal's position, then the animal would feel like it was on stilts and going really quickly. And likewise, if we rotate the objects in the same direction as the animal, it would be it would have to walk further in physical space to go through the same virtual space. And then we can turn off the landmarks, and instead of making the animal forage a certain distance for food, we'll just eavesdrop on the place cells and see what they tell us. So here's the example manipulation. So when the gain is less than 1, that means that the animal experiences less movement in virtual space than it exhibits in physical space. When the gain is equal to 1, the landmarks are stationary. It's going through 1 virtual lap for every physical lap. Let me show that video again. So here the gain is less than 1. So it does, in this case, 1, now 2 physical laps, but during that time, it only went through 1 virtual lap because we were rotating the landmarks as a function of the animal's position. Oh, I guess I already had planned to play this twice, so I didn't need to back up. Forgot that I did that. Okay, so then if I flip that around and I say, let's move the landmarks in the opposite direction of the animal, now it's going to experience a gain greater than 1. It's like super rad. It can, you know, for the same physical walking speed, it's going through virtual environment faster. Okay. So what do we expect to happen? Well, first of all, so I'm going to be showing a graph here where the y-axis is the angular position of the animal, either in the lab frame or the angular position plotted relative to the landmarks, which is a moving frame. And then what we're going to show, and the x-axis is showing laps. So each gray region, gray-white transition is a new lap. So this is 1 lap, 2 laps, 3 laps, 4 laps, and 5 laps. And so you can imagine there's a diagonal, a line here starting from the origin and going up to the top right corner, which would represent, actually I think it's actually starting somewhere in the middle. But anyway, there's a line that's representing the animal's position on this graph. And every time we get a spike, we plop it down here on this graph. So this particular neuron likes to fire when the animal is about 270 degrees in the lab frame and in the landmark frame when the gain is 1. That is to say the lab frame and the landmark frame are actually the same. Okay, so that's the first several laps where we just kept the landmark stationary and we listened in on a place cell. Then we begin to crank up the gain. And what we see is in the lab frame, the firing position begins to drift. But importantly, in the landmark frame, it's still firing in almost the same position, meaning that the place cell is believing our virtual reality and it's spiking whatever the animal is in between these 2 landmarks. Okay? And then we hold the gain constant for many, many laps and give the animal time to recalibrate its path integrator, if that indeed is going to happen, right? And then we do an assay where we then turn off the landmarks. Well, I guess this is for showing the manipulation, but I already showed this basic idea, so I'm going to skip that. Then we do an assay where we turn off the landmarks and watch what happens. But before I do that, let me jump into a technicality, which is really important, which is how do we estimate the rat's internal gain? Well, we can take laps and unwrap them like this: lap 1, lap 2, lap 3, and lap 4. And if the hippocampal gain is 1, then we expect the firing rate. Remember how we can make those color maps? Well, we can do a one-dimensional version of that when it's just running in a circle. We can estimate the firing rate of a particular neuron and plot it as what should be a periodic function where it fires once per lap. If, on the other hand, the hippocampal gain is 0.5, that means that the rat thinks it's going half as fast as it really is. And in that case, the hippocampal gain would only fire once every other physical lap. On the other hand, if the hippocampal gain were 2, then that would mean that the rat's place cells were firing twice every lap. And we can do a very simple Fourier analysis of the firing rate of these individual neurons to estimate the rat's hippocampal gain. I shouldn't say very simple. Ravi Jayakumar has had to work really hard to get this to work in practice. But here's an example spectrogram where the x-axis is laps in the lab frame and the y-axis is the spatial frequency. And you can see there's this very strong harmonic that we can follow for a given place cell, right up to a gain of 3, which was the final gain of our experiment. So we see that the place cells really are sort of following the hippocampal gain. And we can estimate the gain as just the rate of change of the movement in the rat frame of reference, the hippocampal reference frame of reference, dr, to the change dx in the physical space. Okay, so going back to our experiment, this particular place cell fired very stably at 270 degrees in landmark frame. And then after 50 or so laps, we shut off the landmarks. And there's a couple things that could happen. One is the hippocampal gain, which is now drawn here in yellow, that's our estimate based on this Fourier analysis of the firing rates of individual neurons, could go right back to 1. I mean, look, the animal knows that its legs haven't changed length. It's only been doing this for a few minutes. And maybe there's no reason that the hippocampus should recalibrate in such a short amount of time, and it would just go right back to firing in lab coordinates, and the hippocampal gain could go right back to 1. Another possibility is that if we, even though we're turning off the landmarks, if we kept the landmarks going in our own mind and kept the landmark frame of reference, maybe the rat will been so convinced by the virtual reality that we'll have recalibrated its path integrator, and the hippocampal gain will stay at this high value. But what we see is actually something kind of in between, where it's mostly recalibrated. And you can even see that the firing rate does drift in both the lab frame of reference and the landmark frame of reference, but the firing is very consistent with the final value of the hippocampal gain. And the hippocampal gain estimate stays very close to the final value, which in this particular experiment was 1.8, okay? Different than when I showed you the spectrogram before, which was a final gain of 3. In this particular experiment, the gain was 1.8. And so it does indeed appear that this manipulation is causing the animal to sort of recalibrate its internal path integration. And if you plot multiple place cells, another really important sort of sub-finding of this paper is that the place cells continue to fire together and maintain a coherent representation of the animal's movement. We had a blue place, well, they weren't blue, red, and purple or whatever, but we have place cells that we're plotting in blue, red, purple, and green, and they continue to fire in the same order, and they're all recalibrated together. And so that was actually kind of an important piece of evidence that this cognitive map idea, that it's a consistent internal representation and that can be maintained even when the lights are turned off, is true. Okay, so in the interest of time, I won't show all the videos, but here are all of the trials. Each trial is a different day where we did different gain manipulations, and then we follow the gain for up to, in this case, 80 laps after we turned off the landmarks. And one of the really surprising findings is how robust and permanent that gain recalibration is. In the absence of evidence, the hippocampal gain does not keep updating. It took its evidence when the landmarks were on, and then when you turn off the landmarks, it now has remembered that new gain and it maintains for a long period of time. What we did was we plotted two things. What was the gain of the final manipulation? And what was the recalibrated gain immediately after? And we find that across all subjects and across all gains, we see this recalibration plot, which is that all of our RAPs recalibrated. And they recalibrated not completely. If they had recalibrated completely, they would have followed the dashed line, but they slightly under recalibrated, like you might expect. As an engineer, that you're not going to fully recalibrate in that amount of time. And that makes some sense. And then I guess I took this slide out, but then we also plotted the gain immediately after turning off the landmarks and the gain 12 laps later, and saw almost no change. So it's the landmark manipulation recalibrates the path integrator, and that path integration recalibration is at least fairly stable over many laps.
Cool. Thank you so much, Noah, for fantastic talk. Yeah, let's give the most enthusiastic Zoom applause we can. Yeah, exactly. Yeah, so unfortunately, Noah, you do have a meeting in 1 minute, okay, theoretically. So let's do, the highest priority questions, but then I might have to cut it off, and maybe we'll come up with some other ad hoc way to give opportunity to answer more questions. So I got to talk to Noah extensively earlier this morning and last week, or 2 weeks ago. Anybody else have burning questions they want to ask? And you can try to either unmute and talk, or raise your hand, and or type in the chat.
I think in the interest of time, just to speak up. Thanks, Eric.
Okay, well, I'll be selfish. Looking very like the latter work with being able to steer the neural state through manipulation of the sensorium, I think is super exciting. Like really long-term sci-fi future. What kinds of neurologic disorders or pathologies or injuries or things like that could that type of manipulation, scaled up, affect in people? Have you thought along those lines?
Well, I mean, it's not so futuristic, but maybe not as ambitious either. We've got a project with Amy Bastian where, and this is something that was, obviously I'm glad I didn't put it in, I wouldn't have had time, but where we've done manipulations in closed loop where we modify movement output and then modify a virtual display to help people with cerebellar ataxia. So people with cerebellar ataxia tend to be either overshooters or undershooters, and they look like they have a poorly tuned control system. If they're overshooters and you provide them phase leading feedback, that is, you show their hand ahead of where it is in the right way, then you can get them to reach the target much more smoothly. And similarly, if they're undershooters, you actually retard on the screen where their hand appears, and they push more vigorously to get to the goal more effectively. And so that's something that I could imagine becoming, with Google Glass and, some other things, and maybe you wear green gloves or something and make your hands sort of display on your virtual environment in the wrong position, but that's just right to help you improve your movements.
Actually, I could speak to that. We were inspired by those experiments, and we've been doing a series of human virtual reality experiments where we look at proprioceptive gain. So we're actually changing the gain on your visual proprioceptive position of your hand.
Oh, neat.
And we found, very similar to what you've done, that you can induce updates in the gain relationship between the different sensory modalities. But one thing we found that has been raising more questions than answering is that the apparent remapping, the recalibration degree, is contingent on how much you're moving. So if you're only moving a little bit, you're only getting a little bit of error signal, and you're doing a smaller degree of recalibration. And so I was wondering if you had any insight into what might be the cues of this recalibration, because these experiments show very clearly that it happens, but there's not yet like a system model understanding of what mediates it. Like, what are the cues that are used to spur recalibration?
Yeah, it's a really interesting question. It's kind of, there's an explore versus exploit kind of thing where it's sort of you want to do some sort of active sensing, kind of learn how to move better, and learn how to interpret your own internal values better. And I wonder if that could just inform some sort of training paradigm. But I guess it depends on, if the goal is to achieve a certain movement objective, then you needn't necessarily adapt your controller perfectly. And you wouldn't want to make large erroneous movements just to get a better gain in order to achieve that. So I know that in the adaptive control literature, there's lots of results that say you're guaranteed to get to the right end state, but you're not guaranteed to get to the right parameters unless there's a sufficiently exciting input. And this sounds very similar to that.
Yeah, I think it's sort of similar to the uncontrolled manifold type work too. If the error isn't an error, not an error.
Exactly. So, neat question. Okay. Cool, thank you, I'm flattered.
Okay, cool. So I think in the interest of getting Noah to his next scheduled meeting, we'll go ahead and conclude this session. You know, if you have other questions, I won't invite you necessarily to bombard Noah, but bombard me, and I'll help maybe filter them or something like that. Or if you'd like to connect, obviously feel free to reach out. So thanks so much for your attendance, and hope to see you all next week.